Are we okay? Welcome back. We go on this morning session with Jesse Veenvliet, who works at the Max Planck Institute for Molecular Cell Biology and Genetics in Dresden. Jesse.
Thank you, Denis, and especially also thank you for the really kind invitation. I think it's really exciting. This whole meeting is really exciting, and we had some beautiful discussions already yesterday over dinner about the power of these in vitro platforms, essentially. So I call my talk Stembryogenesis, creating to understand, because what we have, I think, been doing in more or less the last 5-6 years, a lot of different labs, and me inclusive, is to create models of embryo development, essentially. And I hope that the next decade is actually going to be the decade where we now are going to take these models and really use them to kind of like understand mammalian development in much more detail, because we will have the power to interrogate it at a kind of like unprecedented ways. So what is Stembryogenesis, and why do I actually call the process of generating these in vitro structures Stembryogenesis? This is because it's a kind of like portmanteau of stem cells and embryogenesis, because what we do in the lab, and what you have heard about this morning, is that we take these pluripotent stem cells and we kind of like in vitro engineer them into organoids that look like an embryo, or at least part of an embryo. And this is, as you know, a very rapidly evolving field. And here I'm showing you some mouse systems that we have to date. Like, small disclaimer, I made these slides a while ago, so they are by no means a complete depiction of the state of the art of the field, but it's just to give you an idea about the things that we can actually do. So we can actually model peri and early post-implantation stages, and here you see these models, so the blastoids that Nicola will talk about later today. We can model peri and early post-implantation stages using these ETX or ETX embryos that essentially not only take embryonic stem cells but combine them with also extra embryonic stem cells. And then, most important for my talk, we can model post-implantation stages, and this was really pioneered by the lab of Afonso Martinez Arias, who generate these structures called gastruloids in as early as 2014, which is basically a adaptation of the classical embryoid body protocol. And then what we have recently shown, or what the lab of Afonso Martinez Arias has shown, and our lab as well, is that we can actually now transform these gastruloids into to have much more complex architecture, much more similar to the embryo. So either you can transform them into a string of somites, or what we showed is that you can actually, by very small manipulation, and I'll come to that, you can now take gastruloids and create these trunk-like structures that now have a neural tube that is flanked by somites. And importantly, this is not just limited to basically recapitulate and reconstituting mouse development, because we can also do this with human pluripotent stem cells. And here are just some recent examples of what we can do. So we can generate essentially neural tube organoids, these somatoids, so two different iterations of the protocol, one without periodic formation of somites and one that has the periodic formation of periodic somite formation that you see in vivo. And then, but these are all like single lineage or single tissue models essentially. And then there are also these multi-lineage models, for example, human blastoids, but also these post-implantation amniotic sac embryoids. We have these MLOs, the elongated multilineage organoids, and of course the human gastruloids. And in our lab, we have been working in the past couple of months on basically a human trunk-like structure model. So this is something that we can now do. So we can generate human gastruloids, but we can also generate human trunk-like structures. But one disclaimer here is that in a lot of cases, actually our human trunk-like structures, they have an overproduction of somites at the expense of neural tube formation, which is actually a condition that in a mouse system we can only generate by changing basically the chemical composition of the medium. So we have these two different classes that we can get at the moment in the lab, but we're on the way to getting like human trunk-like structures. So one thing that you might ask is why actually all this hassle? Why don't you just take the embryo instead of taking an in vitro protocol? And I think most of the people here will appreciate the use of using in vitro platforms. But just to quickly discuss this. So there are, of course, massive limitations of in vivo work, and this has to do with scalability, so we can't get a lot of embryos at the same time. Accessibility, so the embryo is notoriously difficult to basically access. Tractability, so it's very difficult to manipulate the embryo in vivo. And finally, the measurements that we get are typically of a low spatial temporal resolution. Of course, we can generate all these single cell atlases, but this is limited to basically having RNA measurements, for example. But if you think about imaging, it's very, very difficult to get measurements with high spatial temporal resolution. And this means that an integrative understanding is almost impossible. It's very hard to obtain using an in vivo system. For example, like how do mechanical and chemical cues interact to robustly shape an embryo. For human, of course, ethical and legal challenges come on top. So the stages that we are studying in the lab, it would, like in many countries, not even be legal to actually do this in vivo. And in fact, this is a decades-old problem, so it's not something new. We actually know this. Like Alan Turing already said, essentially a very famous quote of his famous chemical base of morphogenesis work, where he says that you essentially have to look at cases where chemical and mechanical information can be uncoupled from each other, because the fact that they are so integrated adds enormously to the difficulty of studying these systems. And at the same time, Jane Oppenheimer, Ben mentioned her this morning, said that the integrative power of the embryos is so pervasive that it should not be overlooked. So we're kind of dealing with a catch-22 problem here. Like on the one hand, we should not overlook this integration of mechanical and chemical cues. But on the other hand, traditionally, we have to confine ourselves to cases where they can be separated because it's so difficult to study their integration. And I think this all comes back to one major question that we have in the lab: how is a mammalian embryo reproducibly shaped? So how are these massive morphological transitions that you see here coordinated with lineage decisions, and how is this basically achieved across spatial and temporal scales? And this is pretty much a problem of emergence, and as Ben put it very nicely this morning, actually also of downward causation. I can't agree more. But also it's a problem of robustness. So how is an embryo always shaped in the same way, and how is the morphological outcome always the same, despite a huge variety, for example, of the cellular microenvironment? And it's not too difficult to see that you can't explain this robustness simply by having like some kind of like genetic program that eventually, via biochemical inputs, leads to morphogenesis, because this would not be a very robust way to generate an embryo. However, a system that would incorporate a lot of feedback loops between basically these morphogenetic information modules, so genes, biochemistry, geometry, mechanics, and where all this feedback would eventually feed into morphogenesis, and where at the same time actually have then this downward causation from morphogenesis to the single cell level, essentially. This would be a very robust system to shape an embryo. Now the problem is that this approach is very attractive if you think about classical ways, classical developmental genetics, where you knock out a gene and you study basically the phenotype. And seeing an embryo as kind of a self-organized system is much more difficult if you really want to interrogate this, because it's very hard to actually interrogate all these feedback loops in a system that is not easy to track and not easy to manipulate, like the mammalian embryo. And this is why we have learned quite a bit about this kind of mechanism from Drosophila, even from zebrafish, but not from mammalian embryos, because they grow in utero and we can't see them. Now, this is the reason that many people, including our lab, have started to sculpt with stem cells. So what we particularly do is that we take embryonic stem cells, we let them aggregate, then we pulse them with a powerful Wnt agonist, and this basically induces them to break symmetry, to elongate, and eventually self-organize their body axis. And this is the gastroloid system that this morning you have seen. So essentially these structures are now, at least at the coarse grain level, they are pretty much patterned like an embryo, or like an embryo without a head, to be more precise, but they lack the stereotypical architecture. So again, these are the gastroloids. So what I have shown is that you can supplement these structures with an ECM in the form of matrigel, and this now pretty much unlocks embryo-like architecture. So now we get structures that are not only patterned like an embryo without a head, but they also are really have the stereotypical architecture. So they essentially morphologically resemble the core part of the embryonic trunk, and we call them therefore trunk-like structures. So just to give you a bit of history about how we arrived there, and at the same time give you the kind of like crash course about axial elongation and lineage decisions in the posterior end of the embryo. So if we now just look at an embryo, we see that we have these neuromesodermal progenitors here. They co-express two transcription factors, SOX2 and brachyury. And these are essentially the axial progenitors that fuel the formation of the body, of the posterior body axis. Now these NMPs, they can upregulate brachyury, and then they take the mesodermal exit route and continue to form somites, or they can upregulate SOX2, and then they will continue to form the neural tube essentially. And this process is very tightly coordinated by morphogen gradients, so opposing gradients of Wnt, FGF, and retinoic acid, as well as a mediolateral gradient of PMP that basically confers a mesodermal subtype identity. Now, to cut a very long story short, when we were essentially looking at the genetics of the system, so where morphogens, at morphogen gradients, at effectors of signaling, at transcription factors, at HOX, we saw that pretty much the genetics of the system are right. And this was, of course, very nicely also shown in the paper by Alfonso Martinez-Arias and Denis. However, we could infer that boundary conditions are not met. So in vivo, we have the extraembryonic tissues, but also some embryonic tissues that, for example, secrete an extracellular matrix that then will confer mechanical and chemical cues that are in vivo very important to shape the embryo. And this was actually very, very easy to test. So what if we would take gastroloids and just look here at this really beautiful segregation of neural in green and mesodermal domains in magenta, but the absence of architecture, and we just supplement them with an ECM in the form of matrigel. So what this now does is that it completely transforms these gastroloids into trunk-like structures. So these structures now have basically somites that would in vivo continue to form cartilage, bone, and skeletal muscle, and they have a neural tube that would in vivo continue to form the spinal cord. And here you see a light sheet rendering imaging of these structures. Then we did a lot of benchmarking to make absolutely sure that our system resembled, to a large extent, the embryo. And one of the things we did was, of course, single-cell RNA sequencing. And here I'm just showing you that we have here these neuromesodermal progenitors, so the actual progenitors, and they are flanked with basically the neural cells. And on the other hand, you see the whole somatic trajectory recapitulated. And we also get some other cell types like endoderm and PGC-like cells. So we have a good reflection of cell states of the embryonic trunk. Moreover, when we compare our single-cell compendium to an extensive compendium of the embryo, we can see that there's a high accordance between the transcriptional cell identities of the TLS and the embryo. And also we see that gene regulatory networks are pretty much conserved, which we can show by pseudo-time analysis and trajectory inference. But what's, of course, or the information that single-cell RNA-seq data is not giving you is if actually these cells are expressed at the right place, or these genes are expressed at the right place at the right time. So we put also a huge effort in actually looking at the localization of marker genes of certain tissues. And here you see this for these neuromesodermal progenitors, that they are nicely at the posterior end. They co-express SOX2 and brachyury. We can computationally infer that they have the highest differentiation potential. And also if you look at selected genes from a somatic gene regulatory network, you see pretty much that the expression in these trunk-like structures kind of like copies that of the embryo. What we see at the cellular level is that basically the addition of matrigel now triggers the induction of apical basal polarity, so the cells now become polarized, and this is essentially not seen in gastroloids. But in the embryo, of course, the cells of the somites and the neural tube, they are apical basally polarized, and this is one thing that we trigger by the addition of matrigel. And also, very excitingly, we can recapitulate more emergent properties in the system. So here I'm showing you the segmentation clock as an example. So the segmentation clock in vivo essentially dictates the periodic deposition of somites, or one pair of somites every 2 hours in mouse. And here you can see that, so here we have a marker for mesodermal cells and their progeny. And what you will see now is that essentially in the middle, the cells, they lose the marker and they become kind of a neural tube. And on the sides, you see these somites like budding off. And if you quantify this, you see that the periodicity is the same as in vivo. One thing that we can finally do with these trunk-like structures is recapitulate in vivo phenotypes. And this is, of course, also very, very exciting. So here I'm showing you, as a proof of principle, the TBX6 null mutant. So this is a very classical mutant already described in the 90s, where what you essentially get in the absence of TBX6 is a transdifferentiation of somatic cells to neural cells, and this leads to the formation of 2 ectopic neural tubes. And this is a phenotype that we can recapitulate with trunk-like structures, so we also get ectopic neural tube-like structures that are derived from cells that essentially wanted to become mesoderm but can't do this in the absence of TBX6. So now, complementary to the embryo, we have these stembryos that are easy to access, track, manipulate, and scale. And this brings me back to the original question that I posed: how is a mammalian embryo reproducibly shaped? And as I said, I think the next decade is going to be a very exciting decade for this field, because I think that we are really starting to learn a lot from these structures instead of just creating them. And in my opinion, there are three main ways that we are using the system to actually learn from it. And the one way that is very nice is that we can actually now generate structures with different morphological complexity, different architectural complexity. So we can generate these gastroloids, we can generate trunk-like structures. They are derived from the same starting structure and only happens in 24 hours actually. And now we can, for example, with live imaging, we can study the difference in formation dynamics of gastroloids and trunk-like structures. And this will basically inform us about morphogenetic information that is necessary to sculpt, in this case, somites and a neural tube. The second way that we can learn from basically stembryo systems is by studying the variation. And this is building on the notion that the embryo is always right and the stembryo is often wrong. So the embryo is always right is, of course, the famous quote from Victor Hamburger, and stembryo is often wrong is my addition. A third way that we can learn from stembryos is by either spiking in at will very precisely in vivo design principles, so cues that we believe to be active in the embryo, or we take them out of the system and we study what basically happens not only transcriptionally but also in terms of morphogenetic processes, cell behaviors, and so on and so forth. So in this perspective in development, we, together with a few labs, we did a kind of like thought experiment, if you want, like what actually happens when you add matrigel to the system. And this will be a cascade of events. It's pretty much a butterfly effect, I would argue, with at the heart of it, like mechanochemical feedback loops. But just to give you a few ideas of how we think about these systems. So if you would basically generate gastroloids, which are like free-floating cultures, then if these secrete morphogens, or if they secrete extracellular matrix proteins, they will float freely into the medium and will basically diffuse out. Now, if we add matrigel, we basically put a constraining factor there that keeps these factors at the surface. So, for example, laminin will bind some ECM proteins that are secreted by the structures, and this will basically constrain them at the structure matrix interface. And the same, of course, accounts for example, for morphogens. Of course, there will also be massive changes in the landscape of stresses and flows, and I'm not going to go into detail for that. What you might also see is that basically the apposition of different tissues becomes different, of signaling and responder tissues due to different flows, and this is something that Ben has referred to as tissue tectonics in one of his perspectives. And of course, we're not only looking at differences at the tissue scale, but this all like goes down to essentially cellular scales, where, for example, differences in the forest landscape may eventually affect the subcellular localization of mechanoproteins, which might then impact cell decision making. But let's just give you one concrete example now. So one thing we noticed when we transcriptionally compared gastroloids and trunk-like structures is an upregulation of morphogenetic regulators and especially integrins. So we saw a very profound and consistent upregulation of integrins, but also coherence in trunk-like structures compared to gastroloids. And as you all well know, integrins actually link the actin filaments to basically the extracellular matrix proteins. However, if we now look at ECM factors, so if you look, for example, at fibronectin, we do not see any transcriptional differences. So yes, basically the anchors, they are, or the integrins, they are differentially expressed, but not the ECM proteins. So this led us to believe, in combination with a whole lot of other observations, that it's not so much about the concentration of ECM proteins that somehow like drives the embryo-like architecture, but it's about the localization. And in particularly, we had reasons to believe, because of other observations, that what matrigel essentially does is that it like traps the nascent ECM that is secreted by the structures at the structure matrix interface, and that this is a very important component in driving the architecture in these systems. So what is the evidence that we actually have for this? I'm going to show you this for fibronectin. So what we see if we compare gastroloids versus trunk-like structures in mouse, in the mouse system, that now indeed fibronectin is confined at the structure matrix interface and also in the somatic cleft, and this is very similar to what you would actually see in the embryo. And here you see how this essentially co-localizes with the integrins. And excitingly, we actually see the same when we compare human gastroloids with human trunk-like structures. So again, accumulation of fibronectin at the structure matrix interface and in the somatic cleft. So now the second part, or the second thing we are interested in, so which is like learning from natural variation in these systems. Because if you read my question closely that I posed, like I did not just ask how is a mammalian embryo shaped, I asked how is a mammalian embryo reproducibly shaped. So we can actually like rephrase this question to how is developmental variability controlled. And this is a beautiful book for those who don't know it. Like I think for this we should go away from the notion that embryonic organoid systems pretty much have to be perfect. And Alexander gave actually some nice examples where you can actually use the fact that these systems are not perfect to learn a great deal about embryo development, essentially. And I'm not the first person to think about this by no means. So I always like to go back to this essay by Pere Alberg, The Logic of Monsters, where what he essentially said, like, what can we learn about actually developmental constraints if we would be able to fully understand how we get like this monstrous constellations during development, so conjoined twins or conjoined fish. And just to point out that he was by then already like thinking about development as a regulative process, obviously, and he was also not the first. So what if we could essentially understand the design principles underlying the deviations from the normal and really like extract information from these abnormal constellations? The problem that Pere Alberg also described there is that he essentially could not study this, so this remained a very high-level theoretical paper, because and one of the problems that he had is imperfection is the outcome of a process, meaning that as soon as you see that you have like conjoined twins, even if it would be legally possible or ethically possible to study this process, this basically is pretty much the end point, and this means that you cannot really understand how something went back in these mechanochemical feedback loops that are at the heart of embryo morphogenesis. And at the same time, the problem is that embryo development is too perfect. So the embryo is a master of robustness. We essentially the embryo is always right, as Victor Hamburger put it. And this is actually where stembryos are very useful. Because if you think about stembryos, or if you think about stembryos and embryos in a state space, essentially, and now let's put at the axis shape, patterning, and cellular complexity, then the embryo actually occupies a very, very small part of the state space, whereas the stembryo occupies a very different and also a much larger part. And this is like kind of like, do you see this as a trick or a treat? Is this like a trick that embryonic organoids want to play with us? And do they try to deceive us? Or is it actually a treat where we can really start to learn from this? And I would argue that this is a treat because, as I pointed out, like these outlier events in the natural embryo, they are very, very, very rare. And nevertheless, we learn from them. We can learn from development if we understand how things go wrong. People have studied like people that are born without a nose, because this learns a great deal about how the nose develops. But these are very, very rare events, and this is mostly, we only get to the genetics of it because there is no way to assess mechanochemical feedback loops, for example. And what we can do with stembryo systems is that we can actually increase the number of outliers. Now, to show you this in real data, this 60% of the structures that we get, or actually 30%, because this is 60% of 50%. They form basically in an embryo-like manner, so we see embryo-like architecture. Then we see the other 40% of the 50% of trunk-like structures that have a unilateral instead of a bilateral body plan. And the other 50%, actually, they do not form a neural tube at all. They do not form somites at all, so they purely consist of a neural tube. And we can not only look at, so this was natural variation, but we can not only look at natural variation, but importantly, we can also increase variation by, for example, changing the boundary conditions of the system. So here I'm going to show you this example where we actually upregulate wind and downregulate BMP signaling. And what now happens is that we create this bunch of Graves phenotype that, remember, in the human system we see as part of the natural variation, actually. And this system has then these hyperepithelialized somites, and now we can actually, by single-cell RNA sequence in this case, go to the roots of this phenotype, at least at the molecular level, where we essentially see in terms of lineage decision-making that we get kind of the opposite of the TBX6 phenotype, where now actually the neuromesodermal progenitors in wild type give rise, of course, to both neural and somatic tissue, but in this chemically modulated situation, basically everything is rerouted towards the somatic trajectory. So all in all, I think we have a great opportunity here to essentially use the fact that stembryos occupy a much larger part of the morphospace to actually study these mechanochemical feedback loops by very precisely perturbing them and actually see at which part of this stembryo morphospace the structures end up. And these are just some examples that I want to give you where we actually already have started to learn from these systems when we think of it in this way. So here you see, for example, of these cardiac gastroloids from Rossi et al., where from the Lutolf lab, that both the genetics and the chemical composition of the medium play into the equation. So yes, we can add these cardiogenic factors and get like the formation of a heart tube, but actually only in stem cells with a genetic background that appear to be competent for forming these cardiac gastroloids, and this coincides with the ability of these gastroloids to form a foregut. And this is actually something that we know from the embryo, that the foregut is very important for cardiac, for heart tube formation. In the same way, as I've shown you, these trunk-like structures are both dependent on basically chemical composition of the medium, as well as adding mechanochemical constraints in the form of matrigel. And then finally, we know from basically the human gastroloids that different genetic lines of different genetic backgrounds need different chemical or different amounts of a chemical signal to reproducibly form gastroloids. And how we can actually study assay phenotypic variation in the lab is by combining live imaging and multiplexed single-cell omics. I'm not going to go into a lot of detail here, but basically the live imaging allows us to look at, for example, patterning, but also at like coarse grain morphogenetic events. And here, for example, see the importance of live imaging, where you get very similar morphological endpoint, but the way that they reach this morphological endpoint, so these are just gastroloids, is very distinct in the two structures, where you get something that we now dub convergent morphogenesis. And what we can now do is take individual structures and actually get their single-cell transcriptomes. And what we then can see by having single structure, single-cell transcriptomes is that they actually, although we think from something that you would not appreciate from bulk single-cell RNA-seq, that there is actually a huge difference between individual structures. So for example, in this chemically modulated condition, you essentially get two classes. One where you have a massive increase of neuromesodermal progenitors, and another class where you have a massive increase of somatic lineages. And also if you look at, for example, endoderm, you see that the endodermal lineage is very variable in terms of its representation across different individual trunk-like structures. And of course, we can also now zoom into individual cell types. Here, for example, neuromesodermal progenitors, and here the population on the left, these are the NMPs in these trunk-like structures. These are the NMPs in these trunk-like structures that are chemically modulated. And you see essentially that they are very different. They appear to have a different Hox code. They appear to have a different expression of morphogens and so on and so forth. So finally, a few words on in vivo design principles, and I want to take hypoxia as an example here. And this is like a more or less like complete story now, and it was a very nice collaboration with two really brilliant scientists, so Aydan Karslioglu and her PhD student Natalia. And what we looked at here is if hypoxia could be a critical modulator of the Stembryo microenvironment. And this was triggered by the knowledge that actually embryos, upon fertilization, they do not grow under atmospheric or oxygen conditions, but they actually grow under like highly hypoxic conditions, so as low as 8% early on, and then around implantation or early post-implantation, it's about 2%. Nevertheless, we routinely culture these stembryos under atmospheric oxygen conditions, so much higher, 21%. So what is the effect of this difference? And the importance here is that especially the stages that we try to model in a Petri dish, you can see that the embryo grows actually under very, very hypoxic conditions. And Aydan, she had a really interesting observation that if you culture just embryonic stem cells under hypoxia, that they induce a transcriptional signature of the primitive streak, essentially. And especially hypoxia induces wind in embryonic stem cells. So we asked if hypoxia might act as a physiological trigger of axial elongation in the absence of exogenous wind. So just to remind you that in a normal situation, in normal gastroloids, their induction of polarization, elongation, and axial organization is dependent on the exogenous supplementation of a very powerful wind agonist that we call chiron. So what we asked if actually hypoxia-induced expression of endogenous wind might be a way to kind of bypass this need for exogenous wind signaling. Now here you see cultures that form anonomoxia or hypoxia in the absence or in the presence of this wind agonist, and of course they both very nicely elongate and have brachyuria at the posterior end. Now, if we basically culture structures under normoxia and we do not add a wind agonist, the structures do not break symmetry, they do not elongate. If we culture them under hypoxia conditions, we can actually trigger axial elongation just by adding hypoxia as a constraint. This is just to show you that concomitantly we see activation of brachyury. And here you essentially see that we can block this elongation by blocking the Wnt pathway. So we really show that it's Wnt dependent, and here you actually see this by live imaging. So you see we get these two classes here under hypoxia, elongating and non-elongating structures, but if you block Wnt under hypoxic conditions, the structures do never ever elongate. What's very interesting here is that actually the way that the structures break symmetry is actually distinct between hypoxic and, or between just hypoxia-induced Wnt and exogenously supplemented Wnt, and in this case it's much more in vivo-like. And just finally, what I want to show you is that hypoxia is not just inducing spontaneous morphogenetic events, but can also improve lineage representation in conventional gastroloids. So gastroloids that are cultured in the presence of an exogenous Wnt signal. If we culture them under hypoxia, we see an overrepresentation of especially midline structures, so nodal cord and gut endoderm, but also of neuromesodermal progenitors. Whereas like here you can actually appreciate that you get also the induction of midline cells without chiron, but you can't sustain a mesodermal lineage. So these are just the quantifications, and here you see the confirmation by immunostainings that really it seems that hypoxia acts as a kind of like developmental canalization factor that increases the robustness of forming endoderm, and here are the quantifications from that. So you go from 25% to 75% efficiency. So then finally, what we can learn about these systems, I always like this like very maybe not mathematically correct, but still very nice equation or a formula by Paul Weiss, thinking about like emergent systems as systems where actually the variation of the system is less than the variation that you observe in the individual components. I think our systems are a great opportunity to actually identify maybe new components or identify how specific components that we spike into the system interrelate with or can reduce the variation or can change the variation and interrelate with different factors, or can reduce the variation maybe of a system as a whole just by adding them in as constraints. And here it greatly helps us that Stembryos increased accessible state space. And what I've shown you specifically is that exogenous ECM can unlock embryo morphogenesis with contributions of the native ECM, and that hypoxia most likely governs developmental canalization, and is essentially one of these like in vivo design principles that we can now, using Stembryo systems, show to decrease the variation of the system. And of course, another question is which individual components of the system is actually regulating. And this is the book that I really recommend you to read. I will not go through the words of caution and just go to my acknowledgement slide, where I want to thank my amazing group, a lot of fantastic group leaders that started at the same time as me, and also Adriano Bolondi, who has embarked on this whole TLS road with me, and just 2 days ago received this very well-deserved summa cum laude as a PhD. And thank you for your attention.